talking about yet another partnership. Settle down, ladies and gents. I know it's exciting being here in the real world. We're another partnership that's enormously important to Genomics England. And that's partnering with commercial companies to make sure that we take data, we take the important donation of their data that has been granted to us by participants, and we turn it into things that diagnostics and treatments that are available to everyone. We're delighted to have Pierre Farmer with us. He's Associate Director of Data Science at Novartis. Pierre, over to you.
Thank you for this very kind introduction. So I'm very excited to be here today to present to you how we at Novartis, we are using this fantastic resource in order to advance our research, development, and also post-marketing activities. And why are we interested in resources like Genomics England? It is that due to the fact that the whole industry, biotech and big pharma, are facing a productivity issue. And this was raised earlier this morning. And to visualize this, there's the EROM curve, which represents the linear relationship between the number of new medicine that is approved by the FDA per $1 billion spent in R&D. And we can see a very steady decrease that is consistent from 1950 all the way to 2010. And it's so consistent that we can even calculate a half-life of this. So every 9 years, there is half of the expected number of compounds that can be registered on the US market per $1 billion. And today, since 2000, the number is less than one compound. So it takes more than 1 billion to put a compound on the market. And this is an issue. And if you look at the internal rate of return for those that are more financially tuned, it is approaching 0%, which is a very big issue. Now, one of the major causes of these costs or lack of productivity, it is really the expensive, the expense linked to project attrition that happens in phase 2 or phase 3. And in phase 2, 56% of failed trials are due to lack of efficacy. So everybody is looking in the industry, can we find markers of what is the best possible target that will lead the highest likelihood of success in a clinical trial? And our colleagues at GSK did a fantastic paper where they have done a retrospective analysis of their pipeline, and they have shown that target indication pairs for which there's genetic evidence, these pairs have twice the likelihood to make it into a successful phase 2. And that attracted a lot of attention in the pharma industry, and this observation was actually confirmed in AstraZeneca and other companies. So we are more and more interested in using genetics in order to select our targets. And shortly after, I came in a conference and I saw a presentation regarding Genomics England, and I realized that having really in-depth whole genome sequencing data that is linked to lifelong electronic health record, not just the case control, but further than that, associated to biobank samples, and also a possibility to reach out to participants through recall consent, this whole value proposition really opens up value creation opportunities across the pipeline, not just for target ID, but also for developing novel modalities, clinical trials, and finding novel indication for marketed drugs. So this is the purpose of my talk today. It's really to present short vignettes on how we are using, since 2 years, Genomics England in order to create value or to advance our project more efficiently. Before we go into these vignettes, I first would like to acknowledge all my colleagues that has worked on GEL that made this presentation possible. I have a lot of gratitude for all this fantastic team of scientists. Now, the first vignette is about target prioritization, target selection. And I think GEL, we will not argue on this, GEL is not the powerhouse of GWAS analysis like other resources can do. However, due to the nature that it's enriched in rare disease and also cancer, disease that are caused by functional mutation within protein, this is a fantastic resource in order to have gene-centric investigation. So if we have a question or target in mind, what we like to do is to list all functional variants, all the gain of function, loss of function, and associate these variants with the very deep phenotypic information that is present in the database in order to further inform on the biology, confirm a target hypothesis, or de-risk a safety-related question. And sometimes we have also a lot of questions regarding what is the mutation prevalence of a given protein in a given disease, and I will show an example out of this. But another application is about modality discovery. And this, we did not anticipate this earlier on, but before we go into this, I will make a little aparté to give you an example of why pharma is interested in rare disease. This was a question that the participant panel asked the pharma industry: why are we investing in research in this case? And it's not rare disease or common disease. These are interlinked because they're affecting the common pathway. The example that I'm presenting here is Ilaris. Ilaris is a registered drug. It is an anti-IL-1 beta, and the team designed this medication with the indication of arthritis. But when the team arrived to the step of being in a phase 1 trial, they correctly anticipated that a negative result in that trial would be extremely difficult to interpret, because we would not know if it's a lack of efficient binding of the IL-1 beta with the antibody, or maybe IL-1 beta is not as involved as we would hope for in arthritis. So the team opted to first try the compound in a rare disease condition. The condition is called CAPS, and this is a disease that is caused by a single mutation or a category mutation in the NLRP3 gene that results in the hyperproduction of IL-1 beta. So these patients have IL-1 beta specific inflammation. Therefore, if the antibody has a clinical efficacy in these rare patients, we are very confident that the MOA is doing exactly what we're doing. So this is an example of mutually supporting R&D. Now, if we go back to modality creation, go back to when we screen compounds, for example, well, when we're using cell lines and we're doing a on-pathway phenotypic screen, the common trick is to introduce loss of function and gain of function in a studied pathway in order to understand what is the consequence on the biological readout of hyper or hypoactive pathway. This really helps us to interpret the screening results. So is my compound activating, inhibiting, or having off-pathway activity? But this is usually typically done in cell lines, and ideally we would like to have our assays to be as biological relevant as possible. And here GEL provides us a great opportunity because we can access a pool of participants that are all phenotype and genotype. Therefore, we can find individuals that have the gain of function or the loss of function of the pathway of interest. And with GEL, we're currently in a very advanced stage in order to recall participants that is of interest, get their biological material, and then we can perform a functional confirmation assay to validate or devalidate the target. And if we wish so, technically, we can transform these cells into iPS and perform either a low or mid-size compound screen, which will find the first chemical matter that will lead to our future drugs. Another aspect of modality discovery is gene therapy. Now, we'll not disclose here the disease nor the gene that is involved, but suffice to know that it's an autosomal dominant disease, and the disease starts, it's a childhood rare disease. So the challenge here is that there's a perfect homology between the target genes across mammals, so this is humans, but all other preclinical model, whether it's mice, rat, or non-human primate. And this brings a big challenge in regards to the PK/PD studies. It's really important to understand if the virus goes into the intended target tissue and express the transgene to a sufficient level. And to do that, we need to distinguish if the protein comes from the virus or if the protein comes from the valid or the wild-type copy of the host. But if there's perfect homology, we cannot distinguish these two types using mass spec. So the team wanted to insert a missense mutation in order to enable this distinction, but we need to be really, really sure that it will not have an impact on the biological function of that protein, because we want to enable the PD aspect of the study. And this is where GEL comes into role, because we have a large collection of individuals that are genotype, and we can find carriers of missense mutation in that target gene that has absolutely no manifestation of the disease and even a broader category of the disease of interest. Therefore, we can be very, very confident that the missense mutation that we have identified are benign. And by doing so, we are enabling PK/PD studies for gene therapy modality. The next step is translational research, and we need to, we can use also GEL in order to build smarter clinical development plan. And the example that I'm presenting here comes from a team that has developed a low molecular weight therapy for indications that are caused by nonsense mutation. So this is a stop sign codon, if you want, that is in the middle of a protein that will stop the translation of the mRNA into a functional protein. And there's over 1,800 diseases that are concerned, but at different level of occurrence. For example, cystic fibrosis, about 5% of these cases are due to nonsense mutation in CFTR, while in Hurler syndrome, this number goes up to 70%. Cancer is also of interest because a lot of tumor suppressor genes are inactivated by nonsense mutation. So the compound acts by directly interacting with the NMD pathway, where the stop codon is replaced by amino acid, and then the translation resumes, generating an almost wild-type protein. And the proof of concept was realized in vivo, but the team received the indication that a multi-indication registration was needed. So the question is, what are other diseases for which the prevalence of nonsense mutation would be of interest for further clinical development? And we have used GEL here in order to mine both the cancer and rare disease cohort. We'll just focus on the disease. We have used in conjunction with other resources to build on each other's strength. And what we observe is very consistent prevalence of nonsense mutation in tumor suppressor genes across these different resources, and also heterogeneity in terms of cancer type. Some cancers, nonsense mutation was way more frequent than others, so we were able to prioritize indication based on this information. Now we're moving to formally into development, and the example we have is an example of precision medicine. Mayzent is a medicine administered to MS patient, multiple sclerosis in Europe, but this medication is not yet registered in the US. Now, the team is preparing the FDA dossier, and in this, the FDA has asked us for a contraindication and dosing companion diagnostic kit. Why? Because there is a very rare variant in which these individuals that are carrier will rapidly metabolize Mayzent to the point that Mayzent will no longer be effective. So we need to exclude this patient. But to build a companion diagnostic kit, we need to have the physical samples in order to build and provide all the validation result that the FDA requires in order to make the approval of the companion diagnostic kit. And finding these DNA samples is not easy. Classically, this would be a population screen, and we calculated that this would take about 25 up to 36,000 of people that we would need to be screened due to the low prevalence of that genotype. However, what we have done a simple query in the GEL database. Within minutes, we have found 90 individuals that carried that genotype. And thanks to GEL, and we are very grateful, we were able to secure access to these samples, and then the team can now build without eliminating the need for a population screen in order to go forward with a companion diagnostic kit. In perspective and development, we're very excited with the genomic medicine service offer that is upcoming for two reasons. Why? It is great to have phenotype patients that are entering the healthcare system that will be sequenced either with whole genome or gene panel with linked lifelong electronic health record. What a great way in order to accelerate the enrollment of clinical trials that are ongoing in the UK, both academic and industrial. And there's another very good use of this data that is generated. It is for synthetic arms, synthetic control arm. The classic way would be to have a two-arm intervention, the standard of care on one side and the interventional arm on the other. But if we could replace the standard of care with all the data that is contained in the electronic health record, that would be a fantastic way to be more cost effective and improve the productivity of the development phase. And this is possible. I'm going into the medical affairs section, but less than a month ago, Novartis received a market authorization for the use of alpelisib, which is a drug that is already in the market, but for a breast cancer indication. But now we received that authorization for a rare disease called PROS, and these patients suffer from a gain-of-function mutation in the PIK3CA gene. And alpelisib, it targets PIK3CA kinase in breast cancer. But what is really nice here is that no clinical trial at all was performed. That registration was done using only available data found in compassionate use of alpelisib in these rare disease patients and removing the need of a trial. And we got that authorization. So it is possible to use real-world data in order to speed up the access of patients that have unmet need. Partnership. So I hope that I provide you some examples of how this close interaction between healthcare, academic, and industry can really benefit all parties. And to maximize, and we truly believe that this has disruptive potential for the industry. And we also need to shape up a little bit how we are organized. And I will take one example, culture and training. So for example, real-world evidence colleagues, they're very familiar with electronic health records, much less familiar with using omics in order to do target ID, for example, and the converse exactly true for our discovery bioinformatics, very familiar with omics, much less with electronic health record. These two live in two different cultures, and we need to bring them closer together. Now, there are a lot also within this group here, opportunities for better partnerships. So what are the opportunities that this community can we bring together in order to enhance GEL capabilities? I will give you an example. So for example, listing all the pain points together, prioritizing them, and co-creating a solution. Currently with GEL, we are co-developing a dashboard that will enable non-data scientists to do some data mining on aggregated data. And that is, of course, will be of benefit for the whole community. And I am sure we can think of other opportunities, like storing derived data from publication. There was a bioinformatician that contacted me last week saying, Pierre, this is a fantastic paper. Can we find all the association results? I said, hey, that's a great suggestion. Maybe we should do that after the day, of course, that the paper is published. In summary, I think that we have shown that GEL is far more than just a genetic data set. For industry, we have enterprise-wide value creation applicability and the importance of partnership. I think I discussed a little bit less, but it is, I think GEL is more at the interface between pharma and healthcare system, and I think this will mutually benefit each other. We can also see GEL being a hub between technology companies. Daphne, for example, is a perfect example, and other academic groups or pharma partners. So I think that the partnership is really key. In conclusion, I will go back to the Urum's law, and there's a great paper by Ringel and colleagues that published that observed that the curve is starting to break. Okay? And they sort of digged more into this, and they provide several reasons, but two that is of strong interest for this group. One is that if we look at the very recent years, there's an enrichment of genetically validated targets that had FDA registration. And these was, for example, in the past 7 years. And second, there has been an increased registration for indication related to rare disease. So I think we can say kudos to GEL, kudos to this community, and if this curve continues upwards, that would be fantastic news. So thank you for your attention. I'm now ready for your questions.
Pierre, thank you so much. And actually, it takes us from thinking about, I mean, if you were thinking about rare diseases as countries in the world, we think of them as kind of, separate little enclaves with really defined borders. But actually, maybe we should be thinking about it in, say, terms of languages spoken. If you put languages spoken overlaid on countries, the languages are the kind of the universal mechanisms that you're talking about.
Yes.
And the countries and we need to break out of thinking of the country of rare diseases as absolutely these bound constituencies, and think about them more in terms of the mechanisms that are jointly seen.
Absolutely. Rare disease is not independent of the common disease.
Yes.
For example, like arthritis or CAPS in the example that I presented, or breast cancer and PROS. There's a lot of overlap.
There's a lot of overlap. Questions from our audience here? I'm going to come to Parker. Can we have a microphone down here? And a question over here too. Parker, any thoughts from you on this very compelling vision from Pierre?
Thank you. Yes, I was particularly inspired by the Mayzent example, and I think it's so powerful that you were able to accelerate an FDA process. Can you think of other examples where this is happening, where we're able to use access to, and not necessarily at Genomics England, but just where you're able to cut straight to a signal that you need by looking at these highly curated populations?
I think that target ID is the one that I think we started there. But I think that we for example, having phenotype and genotype patients to speed up clinical trial, I think that there is possibilities with the GMS service to go into this direction. And also maybe having if we could have ways to query the data in a more privacy-preserving way, that could be a fantastic tool to really have, to keep the privacy issue more tamed and more manageable, I would say. Other aspects, I think real-world evidence data, for example, the PROS is fantastic. Also, it removes the need of a clinical trial, so that is huge for anyone who is interested in curing disease, whether it's academic group or pharma, of course.
And just a quick follow-up, as I know that we are trying to become in the UK a recallable patient resource, what are the kind of operational expectations of companies like Novartis? How quickly do you need to move from identifying a patient cohort to having the blood in a PBMC tube in your labs?
What we would like. So as soon as possible, I think that would be the desire. But I think that mechanism is really, the recallable patients is another great way in order to increase the relevance of our biological assays and to work as close as possible with the disease background and less of more artificial system that cell lines are. I think that would be the...
Pierre, we've got another question I want to get to.
Yeah, great talk, thank you. So you spoke about, first of all, the PROS example, and second about a dashboard, which would be which would allow non-data scientists to data mine.
Yes.
How do you actually encourage and get the patient groups and their champions to actually actively do the data mining and come up with the initiatives?
And a brief answer, if you would, Pierre, of this enormous question.
I think that it's all about data science literacy, and I think that we have as a responsibility as data scientists, we have to teach how everyone can be more familiar with this. That is also true for bench scientists, for example, to be more familiar with data modeling. But the type of analysis that we're thinking is something very simple, like just what is the prevalence of a given mutation in a given indication, and that for many times that's sufficient. Right, so I think that would be start easy, and then we can progress together, and to have the more and more people interested in this.
So Pierre, I'd like to challenge you here and now to work with Genomics England and its participant panel to produce a little course on data science use by patients.
Yeah, yes.
There you are. Quick, write it down before he forgets it. Thank you so much, Pierre. That was terrific.
Thank you.
So we're just going to reset for our next session, and of course, enormously important to make sure that participants know and appreciate the importance of working with industry. Quite often people are scared off by it, but actually they're an essential partner. So, are we ready to start the next session?